First, let us look into initiation of RNA chains. Unlike the initiation of transcription in prokaryotes, eukaryotic RNA polymerases cannot initiate transcription by themselves. All three eukaryotic RNA polymerases require the assistance of protein transcription factors to initiate the synthesis of an RNA chain. These transcription factors must bind to a promoter region in DNA and form an appropriate initiation complex before RNA polymerase will bind and initiate transcription. The common feature of this transcription, however, involves binding of a number of transcription factors to DNA sequences upstream of start point. Here, the structure of a promoter recognized by RNA polymerase II is shown. The TATA and CAAT boxes are located at about the same positions in the promoters of most nuclear genes encoding proteins. The number of transcription factors associated with the initiation process increases as the variety of genes transcribed by the polymerase increases. These transcription factors help in DNA binding of an RNA polymerase to constitute what is described as transcription. After the formation of this complex, initiation of transcription occurs. All non-transcription factors may not necessarily recognize DNA sequences, and a factor may instead recognize another factor or may even recognize RNA polymerase. The initiation of transcription involves the formation of a locally unwound segment of DNA, providing a DNA strand functions as a template for the synthesis of a complementary strand of RNA. The formation of the locally unwound segment of DNA required to initiate transcription involves the interaction of several transcription factors with specific cis acting sequences in the promoter region for the transcription unit. The promoters recognized by RNA polymerase II consist of short conserved elements or modules located upstream from the transcription start site is called TATA box. The position is at +1. It has the consensus sequences TA, TA, AAA reading 5 prime to 3 prime on the template strand and is centered about at position -30. The TATA box plays an important role in positioning the transcription start point. The second conserved element is the CAAT box, which occurs near -80 position and has the consensus sequence GG, CC, AA, T, CT. Two other conserved elements, the GC box consisting of GGG, CGG and octamer box consisting of A triple T, GC, AT is often present in RNA polymerase II promoters. The CAAT, GC and octamer sequences influence the efficiency of a promoter in transcription initiation. The initiation of transcription by RNA polymerase II requires the assistance of several base cell transcription factors. These transcription factors must interact with the promoters in the correct sequence to initiate transcription. The initiation of transcription in eukaryotes begins with the binding of TF2D to the TATA box. The key initial event in initiation is the recognition of the TATA box by the TBP TATA box binding protein, a 30 kilodalton component of the 700 kilodalton TF2D complex. TBP is a saddle shaped protein consisting of two domains which are similar in structure. The Tata box of DNA double helix binds to the concave surface of TBP and induces large conformational changes in the bound DNA. The DNA is substantially unwound to widen its minor groove, enabling extensive contact of TBP. 4 phenylalanine residues are intercalated between base pairs of the Tata box. Next, TF2A joins to the complex, followed by TF2B. TF2F, an ATP dependent helicase, first associates with RNA polymerase II, and then TF2F and RNA polymerase II joins the transcription initiation complex. TF2F contains 2 subunits which has DNA unwinding activity. Thus, TF2F probably catalyzes the localized unwinding of the DNA double helix required to initiate transcription. TF2E join the other factors to form a complex called basal transcription apparatus. 2 other factors, TF2H and TF2J, join the complex after TF2E, but their locations in the complex are unknown. TF2H contain an enzymatic activity that phosphorylates RNA polymerase II. One hypothesis is that the phosphorylation of RNA polymerase II by TF2F releases the enzyme from the initiation complex at the promoter, so that it can move downstream and mediate RNA chain extension. Similar promoter complex or transcription factors are required to initiate transcription by RNA polymerase I and polymerase III. RNA polymerase I and polymerase III use some of the same transcription factors as polymerase II, and some factors are specific to each enzyme. The promoters of genes transcribed by polymerase I and polymerase III are quite different from the promoters utilized by RNA polymerase II.